Hello everyone. Today you are welcome here in cell division. So we will learn about the cell division. We already learned about cell structures and its functions. So today here with cell division. Introduction to the cell division. It is the basic unit of life is the cell. Two main types, mitosis and meiosis. So why cells are divided? Because growth in multicellular organisms, repair damaged or dead cells, reproduction in living beings. Importance of the cell division, continuity of life, formation of new cells, maintains proper tissue and organ functions. Types of cell division, mitosis, body cells, somatic. Meiosis, reproductive or germ cells that have different purposes and different outcomes. What is a cell cycle? Sequence of events in cell life that prepares cell for division and it repeats continuously in living cells. We have here phases of cell cycle. Interphase, that is the preparation stage. Mitotic phase is the division stage, and the order ensures proper cell function. Interphase. Interphase is the longest phase of the cell cycle, during which the cell grows, performs normal functions, and prepares for cell division. Mitosis or meiosis. Although it's not a part of mitosis itself, it's crucial because it sets up the cell for proper division. So the key points to explain here is, the cell is very active even though it does not divide yet. DNA is replicated to ensure the new cells have identical genetic material. Organelles and proteins needed for division are synthesized. Okay, now see the stages of interphase. Interphase is divided into three main stages. G1 phase, gap 1 phase. The cell grows in size, proteins and organelles are synthesized. The cell performs its normal functions like a liver cell producing enzymes. Next is S phase, synthesis phase. DNA is replicated, so each chromosome now consists of two sister chromatids. Ensures that after division, both daughter cells get identical DNA. Next is G2 phase, gap 2 phase. What happens here? The cell continues to grow and produce proteins. Organelles needed for division like centrioles are replicated. The cell checks for DNA errors and make repairs. So this is the final preparation stage before mitosis begins. Alright. Now we will talk about mitosis. Mitosis is the process by which a single cell divides to produce two genetically identical daughter cells, each with the same number of chromosomes as the original cells. So the key points here are, it occurs in somatic body cells, ensures growth, repair and replacement of cells, maintains chromosome number across generations of cells. So think of mitosis as making an exact photocopy of a cell, including its DNA. So stages of mitosis is divided into four main stages, sometimes including prophase, metaphase, anaphase, telophase, plus cytokinesis, division of cytoplasm. So first we will see prophase. What happens here? Chromatin condenses into visible chromosomes. So each chromosome has two sister chromatids joined at the centromere. The nuclear membrane starts breaking down, spindle fibers begins to form. So the key point here is chromosomes become visible and the cell starts preparing to move them. Next is metaphase. What happens here? Chromosomes line up at the center of the cell. The metaphase plate, spindle fibers attach to the centromere of each chromosome. The key point here is, this alignment ensures even separation of chromosomes. Next, anaphase. What happens here? Sister chromatids are pulled apart by spindle fibers toward opposite poles of the cell. Each chromatid is now considered an individual chromosome. So chromosomes are equally divided to ensure identical daughter cells. What happens in telophase? Chromosomes reach the poles and start to decondense into chromatin. Nuclear membrane reforms around each set of chromosomes. Spindle fibers disappear. So the key point here is, the cell is almost ready to split into two. Now we will talk about the cytokinesis, not always counted as a mitosis stage. What happens here? Cytoplasm divides, forming two separate daughter cells. So each daughter cell has the same number of chromosomes as the parent. So the key point here is, this completes cell division. Okay. So there is a quick memory tip. You can memorize the stages as PMAT. Please make another T. P means prophase, M metaphase, A anaphase, T is telophase. Alright. Now, importance of mitosis. Mitosis is essential for growth, repair, and maintenance of an organism. So, key points here for the growth are: helps an organism grow by increasing the number of cells. For example, human child grows because body cells divide by mitosis. Repair replaces damaged or worn out cells. Example, skin heals after a cut. Liver cells regenerate. Third is maintenance. Keeps tissues and organs functioning properly by replacing old cells. Asexual reproduction in some organisms. Single-celled organisms like amoeba reproduce by mitosis. So the key point here is mitosis ensures genetically identical cells for stability in body functions. Next we will talk about meiosis. Okay, meiosis is a type of cell division that reduces the chromosome number by half, producing 4 genetically different daughter cells. So the key points here are: occurs in gametes, sperm and egg cells, ensures genetic diversity in offspring. Chromosome number is halved so that after fertilization the zygote has the correct number. Okay, think of meiosis as mixing 2 decks of cards and then splitting them. Every child gets a unique combination. So some stages of meiosis are: meiosis occurs in 2 divisions, meiosis first and meiosis second, each with its own stages. First, we will talk about meiosis first, the reduction division. Reduces chromosome under from diploid to haploid. Prophase 1. Chromosomes condense, homologous chromosomes pair up forming tetrads. Crossing over occurs, genetic variation, nuclear membrane disappears. Metaphase 1. Tetrads line up at the metaphase plate. Independent assortment occurs, random distribution of chromosomes. Anaphase 1, the homologous chromosomes separate and move to opposite poles. Sister chromatids remain together. In telophase and cytokinesis, nuclear membrane may reform and varies in species. Cytoplasm divides 2 haploid cells. In meiosis second, similar to mitosis, separates sister chromatids. Prophase 2, chromosomes condense, nuclear membrane disappears, spindle fibers form. Metaphase 2, chromosomes line up individually at the metaphase plate. Anaphase 2, sister chromatids separate and move to opposite poles. Telophase 2 and cytokinesis, nuclear membranes reform, cytoplasm divide, 4 haploid daughter cells, each genetically different. Alright? Now the importance of meiosis. Meiosis is crucial for sexual reproduction and genetic diversity. So the key points here are: reduction of chromosome number, halves the chromosome number in gametes, ensures that after fertilization the zygote has the correct chromosome number. Second is genetic variation. Crossing over in prophase 1 and independent assortment in metaphase 1, unique combinations of genes, so leads to variation in offspring, which is important for evolution adaptation. Formation of gametes produces sperm and egg cells in animals, pollen and ovules in plants, prevents chromosome doubling, maintains stable chromosome numbers across the generations. So the key point here is: without meiosis, sexual reproduction would not be possible and offspring would not be genetically diverse. So there is a small comparison here in mitosis and meiosis. In types of cell in mitosis, somatic or body cells. In meiosis, gametes, sperm or egg. Number of divisions in mitosis is 1. Meiosis 2. Number of daughter cells in mitosis 2, and in meiosis 4. Chromosome number in mitosis same as parent, and meiosis half of parent. Genetic variation in mitosis? No genetic variation. In meiosis, yes, crossing over, independent assortment. Functions in mitosis: growth, repair, asexual reproduction. In meiosis, sexual reproduction, genetic diversity. Phases in mitosis: prophase, metaphase, anaphase, then telophase. In meiosis 1, prophase 1, telophase 1, plus meiosis second, prophase second, then telophase second. Alright. So here is a practice set given. You all can go through one by one to all the questions and answers. Okay, see you in the next session.